Hi, Assalamualaikum. In this lecture, we're going to talk about endocytosis. Okay, endo here means inside. So cytosis means cell. So inside the cell. So basically, we are transporting material from outside into the cells. Okay. So we know that plasma membrane is held together by weak hydrophobic forces between the fatty acid tails. So this is our phospholipid bilayer. So you can see, so these phospholipid bilayers, they are basically held together by these hydrophobic forces, and these forces basically gives the membrane its fluidity and its flexibility so that it can move around quite freely. All right. This property or criterion also allows they can basically perform spontaneous breaking and reformation. So this is quite important because in order for them to basically start to produce a vesicle when they are transporting materials in and out of the cells without having to basically cross the plasma membrane. Okay, so what is endocytosis? Basically, you want to allow them an entrance into the cell without having to cross the plasma membrane, and it involves a invagination of the membrane. So basically, the membrane, okay, as you can see here, they start to break and they start to reform. Okay, you have breakage and you have to reformation, right? So the membrane, plasma membrane, our phospholipid bilayer, they start to basically break and then they reform again as they are producing the vesicle. So invagination here is quite important role. So what does it mean by this? It means that it's the membrane here. Okay, for example, here you can see here I'm drawing it here. So the membrane is basically folding inwards. Okay, this is the materials that they want to take in. So as the material get closer to it, so it start to invaginate, start to like being pinched inward. You understand? So they're being pinched inward, folded inward. All right, and eventually this part of the plasma membrane, this part, they start to join together. So here they start to break and then they start to reform. Eventually they split off and they now become the transport vesicles containing the material. I'm going to show you the GIF later. Okay, so the invagination then seal off, all right, basically break off, and the phospholipid bilayer break off and then they start to reform again and they form the vesicle. Two types of endocytosis basically. Okay, we have phagocytosis and we have pinocytosis. So phago is actually a Greek word for eat. Pino is actually for drinking. Phagocytosis is the process where the cell takes in solid substances to be ingested. So basically these solid substance are food, and they are, after being taken up, right, and they are temporarily stored in the vesicle and they will be transported to lysosome for further digestion. Right, pinocytosis is when you have liquids or you have dissolved substances and they're being ingested, and this is basically why they're using it, because this to allow faster entry rather than using protein channel. Might as well just use pinocytosis, much faster. Okay, so as you can see, this is probably an example of white blood cell. So white blood cells basically taken up an organism, a bacteria or virus, whatever. In this case, I'm sure that is a bacteria. So they are being taken up. Okay, as you can see, the plasma membranes start to invaginate and eventually they join up together and they're going to form the vesicle. Meanwhile, okay, the bottom diagram over here, you can see there's a dissolved substances, right? And they are accumulated in the invaginated plasma membrane. Eventually, it join up together and form the vesicle. So can you see from the GIF, all right? So you can see how does the plasma membrane basically invaginate inwards, so, and eventually they form the plasma. They form the vesicle.
See ya!